And with that, I'm going to ask Dr. Drew to stay on because she's going to be presenting our last session of the evening, the NEOCATS Study, a potential paradigm shift for first-line treatment of high-grade serous ovarian cancer. So Dr. Drew has played a key role in both the preclinical and clinical development of the PARP inhibitor rucaparib in ovarian cancer. Her research interests include the targeting of DNA repair pathways as treatment for gynecological cancers and PARP inhibitor resistance. Interviewing Dr. Drew this evening, we have patient partner in research, Linda Brown, and also back with us is Dr. Alicia Tone. Linda was diagnosed with ovarian cancer in 2018 and is an active member of the patient partners and research team. So Dr. Drew, I'll turn it over to you.
Great, thank you. I'll just try and share my screen. So if you let me know when you can see slides.
Yes, we can see it.
Yep, can you see that? And you can hear me okay?
Yes, all good.
Great. So I just want to thank you for inviting me tonight. And as has already been said, I'm going to talk to you about the NEOCATS trial, which is a neoadjuvant olaparib combination ovarian cancer targeted study. And before I sort of do that, I just want to introduce the topic with a couple of slides to try and sort of talk about why I want to do this trial and obviously the trial itself and some of the details. So I shared this recently at a previous presentation last week, and this is a quote from one of my patients in clinic who said to me, "Are you still using carboplatin and paclitaxel, Dr. Drew? My mum had that for her ovarian cancer 20 years ago." And obviously, you know, my reaction was, well, yes, it works and nothing beats it. And we've always done it this way. But essentially, the backbone of, you know, first-line treatment for women with stage 3 and 4 high-grade serous ovarian cancer is chemotherapy and remains chemotherapy. And many women are receiving chemotherapy in the first-line setting prior to surgery, so what we call neoadjuvant treatment. And that might be because the cancer is too advanced to be operated on. It might be because the patient is not fit enough at that point for surgery, or even due to delays in accessing surgery due to COVID and other sort of challenges following the pandemic. And the first line treatment, as we know, is carboplatin and paclitaxel, and some women get that with Bevacizumab, and it's given every 3 weeks, and we usually give 6 cycles of chemotherapy. And if you're having surgery after chemotherapy, it's usually sandwiched somewhere in the middle. But how good is chemotherapy really? So these are the sort of, this is the reality of the outcomes of chemotherapy for women that kind of get chemotherapy first. So the response rate from a recent trial, the ICON-8 trial, showed us that the response rate to chemotherapy was 61%. So response rate means what's the chance of the cancer shrinking on the scans. That doesn't include it staying the same. This is just patients whose cancers shrink with the treatment. When women have completed chemotherapy, on average, the progression-free survival, so the time for the cancer to return, is about 13.8 months, and that's after first-line treatment. And we know that 80% of women who get neoadjuvant chemotherapy, so chemotherapy first because their disease is too advanced, will recur and subsequently die of treatment. So this is, you know, showing that our first-line treatment is not that effective. Now, we have made great progress, and we've heard a lot about this tonight, looking at one targeted type of treatment, which is PARP inhibitors in selected patients. And what I'm talking about there is PARP inhibitors, which obviously we started out using them in the recurrent setting as upfront treatments themselves or as maintenance treatments, and then we've moved on to using them in the first line setting. And we've heard about Solo 1 and the PRIMA trial, and most recently ASCO just presented was the data for rucaparib, again showing a benefit in maintenance, and the benefit is the greatest in those women that have a BRCA mutation or this type of ovarian cancer that has homologous recombination deficiency, which includes those with BRCA mutations. And you can test that by biomarkers such as this thing called LOH-high or the Myriad MyChoice assay. And also platinum sensitivity is an indicator that you might have a homologous recombination deficient cancer. And this is the data from the Solo 1 trial, and this is fantastic. But obviously, if you turn this on its head, you can see that the women at this point, we have 48% of women who've not recurred, which means that 52% of women have. So even with our best sort of first line treatment, we know that we need to do better. So are there any alternatives to chemotherapy that can be explored? And one of the things that I've been working on in a trial that Dr. Lee mentioned is the Mediola Trial. And this is combining different targeted treatments without chemotherapy. And there is a lot of evidence in sort of laboratories and in sort of mouse models and in early clinical trials that these three drugs could work well together to enhance each other's activity on the cancer. And so I, with AstraZeneca, took part in this study called the Mediola Trial, and there are lots of different parts of the trial, and this is just one of the parts of the trial. So in this trial, and this trial completed a number of years ago now, we took women who had not got a germline BRCA mutation, so they weren't women who had hereditary ovarian cancer, and they couldn't have had a PARP inhibitor or an immunotherapy treatment, and they had to have received less than 2 prior lines of chemotherapy, but they all had recurrent ovarian cancer, and they all could have gone on and had chemotherapy. But instead of doing that, they were recruited to the study. And we offered women 2 different treatments, and this isn't a randomized clinical trial, so these 2 cohorts of patients were recruited sort of sequentially. So women could have this triplet combination of olaparib, durvalumab, and bevacizumab, and I've just put on the right here what these drugs are, just to remind you. And then they could have had a doublet treatment of the olaparib and the durvalumab, so that's the treatment without the bevacizumab. And we were looking in this trial, it's a small study, and we were looking for what is the chance of this working, and is this are these triplet or doublet combinations safe? So I'm just going to show you the data for the triplet here, because that's what's being taken forward. So this is called a waterfall plot. I'll just go back one. And I don't know if you're familiar with looking at these, but each green bar here represents a patient's cancer responding to treatment. Anything that goes below 0 means that it's shrunk. Anything that goes above 0 means that it hasn't shrunk. And to be in response, you need to have a 30% or greater shrinkage. So if you look at this waterfall plot here, you can see that most women had a really good response to this chemotherapy sparing triplet treatment. So the response rate was 87.1%. And the length of that response was 11.1 months. But these are women with recurrent disease. So this is not a first-line study. And we've submitted the long-term follow-up of these women to the ESMO conference, which will be held later this year. So I can't present that data to you today. So I said at the beginning that these were women that didn't have germline BRCA mutated ovarian cancer, but they might have had some other genetic abnormalities in their cancer. And we did this test called a genomic instability test on the cancer, which is similar to a HRD test. And we grouped the patient's response by whether they were GIS positive, so they had genomic instability, or they didn't. And what was interesting about the study is that regardless of the result of this test, women were responding. So this treatment seems to work across the board, whether you have homologous recombination deficiency or genomic instability or not. So that's interesting. And the other side of the study, and I'm not going to present all the safety data today, but the summary of the safety data is that this triplet was safe, and there wasn't really any overlapping toxicity between these 3 drugs, and we didn't see any compounding of toxicity. So we saw what we would expect from these drugs as single agents, in the study, and most women were able to tolerate the treatment. So moving into the sort of neoadjuvant setting, which is the first line treatment, we've got a number of studies trying to look at sort of targeted PARP inhibitor-based treatments. And there are a few studies ongoing now in the US. One is looking at olaparib, and the other is looking at talazoparib, and that's another PARP inhibitor, which isn't currently licensed or approved in ovarian cancer, but is approved in breast cancer. And these specific studies are looking at BRCA mutant or abnormal, ovarian cancer, or those with homologous recombination deficiency. So these are very specific studies, and they're very small, and we don't have the results of those yet. And this is the study that I have proposed and has recently been funded by Ovarian Cancer Canada and is supported by AstraZeneca. So this is the study of this triplet combination, and it's going to be investigated in women with advanced stage 4 high-grade serous ovarian cancer in a chemotherapy sparing approach. And there have been lots of patient partners, which I'm very grateful for, involved in the study. I've named the groups there. And also the study is supported with a big translational program and some collaborators I've listed on the top here, such as Connexia Health and Illumina Canada. And the aim of the study is to run it through the Princess Margaret Consortium through Toronto with Dr. Ami Oza's support, but hopefully to have the study open across Canada so that there could be the opportunity for many women to participate. Now, in terms of the study itself, what we're going to try to do is recruit women even before diagnosis. So if you have suspected stage 4 high-grade serous ovarian cancer, you may be able to consent to going into the study and having your diagnostic biopsy, but also the research biopsies as part of the trial. So it won't be just for women who've got confirmed disease, it will be for women with suspected disease, and that will be done as part of the sort of screening progress. And the reason to do this is that we're going to need additional biopsies from women, and we don't want people to have to go back and have a biopsy as part of diagnosis. If it could be done at the same time, that's being proposed. In terms of what's going to happen to all of these samples, so in the study, we're hoping to collect biopsies, we're hoping to collect fluid, ascites, pleural fluid, and then obviously women that go on to have surgery will be able to collect multiple samples through their surgical procedure. And these are just some of the things that we're going to do, but obviously I'm not going to go into detail with all of this tonight. The trial itself, this is what it's going to look like. So it would be for women with stage 4 disease who are planned for neoadjuvant chemotherapy and planned for interval debulking surgery. And we'd be asking these women to come into the study instead of having their chemotherapy. Now, this could be a risky thing to do because we know that we have a standard of care. So we're deviating from the standard of care. So it's important in the trial to be on the lookout early for those women that aren't responding. So after 2 cycles of treatment, women will have a scan. And if they're not sufficiently responding, and that will be decided by their own oncology team and on their scans, then they will come off this trial and go on to standard of care chemotherapy. Those women that are responding can continue with this triplet up until going for surgery. And then because this is a neoadjuvant study, everything that happens after surgery will revert back to standard of care. So the first thing that we're trying to show in this trial is can we not give chemotherapy in this setting, and is it safe, and how effective is it? So it's a small study that will involve up to 40 women. So there are some challenges ahead. So like all clinical trials, they are proposed by us as investigators, and we have to get funding for them. And we do have some challenges still with the funding for the trial. So Bevacizumab is not a drug that AstraZeneca own, and they are not supporting that as part of the study. So they're going to provide olaparib and durvalumab for free, which is great, but we have to fund the cost for the Bevacizumab. And there's obviously additional funds that we're still trying to raise for administrative support. So we have a lot of funding from Ovarian Cancer Canada, but there is still a shortfall in the funding for the trial. So I want to thank you all for listening. And as I say, I thank you to the patient partners, to Linda, who's going to grill me now on all of this. And I just thank you for having me this evening. And I'll stop sharing so that you can see everybody else.
Linda, you're on mute.
Yeah, sorry about that. So Dr. Drew, just before we start, I wanted to say that after watching some of your seminars and videos online and seeing your very extensive and significant list of research publications, how fortunate we are as Canadian women living with ovarian cancer to have had you move from the UK to Canada and bring with you your extensive and inspiring gynecologic research and expertise here. It's extraordinary, and we're grateful. So thank you so much.
Thank you, Linda. I mean, I have to say you have probably Brexit and Boris Johnson to thank for that. but also the team here in Vancouver is a fantastic team, and I feel very privileged to be part of it now. So
yeah, so we're very fortunate to have you. So thank you. So I guess the first question would be, can you explain the current standard of care for women with newly diagnosed stage 4 or high-grade serous ovarian cancer, as opposed to stage 3, and why only women with stage 4 are at this point going to be in the NEOCATS study?
Yeah, so really good question. So the standard of care for women with stage 4 disease, it isn't particularly different to stage 3, although a lot of women with stage 4 disease have chemotherapy first and don't go and have surgery first, because obviously stage 4 means the cancer is more advanced and therefore may not be amenable to surgery. And the other thing that these women can access is they can access Bevacizumab in combination with their chemotherapy if they have stage 4 disease, whereas those women with stage 3 at that point can't. So there are some subtle differences, but the kind of pathway is very similar. Now, the reason why we chose to focus just on stage 4 patients for this trial is there's a number of reasons. Firstly, those women with stage 4 disease have the worst outcomes currently. So they have the greatest unmet need, in terms of improving treatment. But those women with stage 4 disease also have potentially disease that may be more accessible for biopsying. So they might have lymph node, metastases. They might have, other disease elsewhere that we can potentially biopsy as part of the study. And the other reason is that women with stage 3 disease in some centers might get surgery first, whereas in other centers they might not. And it's a bit more of a mixed group of patients. So we wanted to try to focus the study on a specific group so that we could make sure that there was kind of, homogeneity in the patients going into the study. So there's just a number of reasons. But in theory, where this study goes, if it's shown to be successful, it could be applicable to all women, with stage 3 or 4 disease.
That's terrific.
Hi, Dr. Drew. So you explain you call your study a proof of concept study, which I think you should explain what that means, perhaps. And you're measuring safety, feasibility, and fitness for surgery. So perhaps you could elaborate on those things, especially how you're monitoring safety, and what you mean by fitness for surgery as well.
Yeah, so I mean, proof of concept usually is applied to a study where it's the first of its kind. So when we first developed PARP inhibitors, the first ever trial in a BRCA patient was called a proof of concept study, because it was like, do we even know if this is going to work? So although we have the evidence in women with recurrent disease that this is an effective treatment, that was a small study. So this isn't a 1,000-patient study. That the Mediola trial, these cohorts of women were very small. So we have to prove that this concept is applicable to women in first-line settings. So that's why it's called a proof of concept study. And obviously, where there is a standard of care, it's very important that you protect patients from any harm. So what we want to do is focus on safety. And the first 10 patients that go through the study, their data and their results and all of the side effects will be analysed by a kind of independent committee, which will be formed to kind of monitor the progress of the study. And if there's any concern about safety, like there's too many side effects, or there's some concerns about people having surgery after this treatment, because that's what we don't know, how safe is surgery after these treatments, then we would want to stop the study early if there was a safety concern. Now, we know that giving Bevacizumab before surgery is safe, but you have to stop the Bevacizumab a number of weeks before surgery. So that will be happening as part of the trial. And we know that there's a study already giving a type of immunotherapy called pembrolizumab before surgery. And that study showed that it was safe. So, but what we don't know is the triplet, the 3 drugs together, is that going to be safe? So we have to make sure that we are kind of very early on making a decision about whether we recruit the rest of the patients to the study.
Linda, do you want to go ahead?
Yeah, and maybe we answered this question when we were talking about PARP inhibitors earlier. I'm not sure. So stop me, Dr. Drew, if I'm repeating myself. So your trial includes the use of a PARP inhibitor at the very beginning of the treatment journey prior to chemotherapy and surgery. So are you concerned at all that that might lead to some kind of PARP resistance, looking at the treatment journey down the road after surgery and chemo?
Yeah, so it's a good question. So it's unlikely that this study would do that, because this study is just looking at 3 cycles of treatment before surgery. So it's a limited course. And then most of these women will be able to go on to their maintenance PARP inhibitor afterwards as part of standard of care. So I think it wouldn't be this study that would potentially cause resistance, but what we're actually trying to do is, which is the sort of opposite of that, is that we think, and we don't know this, that maybe the cancer is more sensitive to PARP inhibitors before chemotherapy. Now, we don't know this because it's not been shown in a trial yet, but there is some sort of scientific data to suggest that potentially it might work better if it's given first. But that's one of the things that the study's looking at.
That's exciting. Wow.
So Dr. Drew, obviously as one of the funders of this study, we're very excited about it and want it to be successful. So if it is successful, what would be the next step, and what is kind of the ultimate goal that you're working towards?
Yeah, so I guess the evidence from the Mediola trial, shows that this triplet is very effective, and it's more effective than we would think of that chemotherapy would be as effective. But the Mediola trial didn't randomize anybody to chemotherapy, so it didn't compare in the same group of patients, how effective chemotherapy is. So it would be wrong of me to say this treatment is better than chemotherapy. So obviously what we're trying to do is introduce the idea of changing the paradigm and not giving chemotherapy with this small study. And if it is successful, then we would want to do a randomized controlled trial, comparing the triplet targeted treatment to chemotherapy. And that would need to be a much bigger study, and that would involve a lot more patients, and that would cost a lot more. So the first thing is to prove something small, and then hopefully expand it into a bigger study. So this trial in itself will not change standard of care, but it could be a step towards that.
So I think Steph is going to come on and has some questions, unless Linda, you have one you want to ask.
No,
Steph,
I just wanted to comment that it's such a huge paradigm shift. It's extraordinary and so very interesting.
Sorry, I was just going to close my blinds there. The sun is shining in here in Saskatchewan. We do have a couple of questions, Dr. Drew. So the first one here is, from what I recall, Bevacizumab can affect bowel wall integrity. Would this not increase risk complications of bowel complications during or post-surgery?
Yep, so that's a good question. So we already give Bevacizumab, as I say, as standard of care for this patient group. And we wouldn't give Bevacizumab if we were concerned about the risk of what's called bowel perforation or fistula formation. So we already know who those kind of patients are, and those patients would have to be excluded from the study, but they would be excluded from Bevacizumab as part of standard of care as well. So there's a specific things that we look at on the scans, to decide if somebody is at risk of complications of Bevacizumab. But it is certainly something that will be part of the safety, of the study. So for example, if we saw more of that in this study than what we would expect with sort of standard of care, then that would be a red flag for the study. But we would be excluding women who we think are at risk of Bevacizumab bowel side effects.
So on that, then another question just popped up here. So who would those patients at risk be?
So it's generally, so there isn't a sort of patient that you can define by the patient characteristics. It's more about the scan and where the cancer is. So if the cancer is very sort of matted onto the small bowel, or like, very much stuck on the bowel, there is a risk that you could get a perforation or a fistula. And these things just generally happen, or they don't happen. So it could be like it doesn't happen, or if it happens, it's a serious side effect. So it isn't something that the dose of Bevacizumab, or you can't sort of protect patients from this if you give these drugs. So when we look at the scans, we normally discuss with radiologists and obviously surgeons whether we think this patient is at risk, and we would or wouldn't give them Bevacizumab, and that happens routinely anyway. So anybody who was going into NEOCATS, if their own oncologist thought they were at risk of Bevacizumab side effects, this side effect, then they wouldn't be going into the study.
Okay, and I just want to say thank you for so clearly answering all of the questions. I certainly appreciate it, and I'm sure the audience does as well. So thank you. And there's one final question. Are there any preliminary findings from the Athena trial?
Yep, so the Athena trial obviously was a trial a bit like the Prima trial. So it was looking at women who had stage 3 or 4 high-grade serous ovarian cancer, and they had their first-line chemotherapy, after surgery, and then they obviously went on to a PARP inhibitor, which this time it was rucaparib, and again it was against placebo. And the data that's just been presented at ASCO is the data for the BRCA mutation, carriers. So, and that shows, just like Solo 1 and just like Prima, that there is a benefit to having rucaparib. So it was the HRD and the BRCA patients. So it doubled what's called the progression-free survival. So the time it took to the cancer to come back was doubled. So it's kind of showing us what we already know. And these trials, they're all designed at different times, by different companies. And rucaparib was actually the first PARP inhibitor ever to be given to anybody in the world. But obviously it came in slightly later in terms of the ovarian cancer trials. So that's why we're seeing it reported now. But Athena also has some other arms, which is the immunotherapy treatment, and that data hasn't been reported yet. So we only got the rucaparib maintenance data at ASCO just last week.
Okay, excellent. Thank you. So that is
and I
all
think
of the
I would
questions.
just say, actually, Stephanie, sorry, I just think it's important.
Yeah, no worries.
And Anna said this before, and I think it's important to say it again. We don't consider as oncologists one is better than the other in terms of PARP inhibitors. They all seem to perform well. They've never been compared directly with each other in a trial, and they probably never will be. So I think it's important, if you're on a PARP inhibitor, not to think, oh, there's a new one now and I want to go on that if you're getting a response to one, you're likely to get a response to the others. And if you don't respond to one, you're not likely to respond to the others. And it's just about what's approved and what's available. And I think that's the most important thing rather than which one you're on.
Thank you for clarifying that. I'm sure a lot of people had that question, so thank you. And that brings us to a close. We have no more questions. So Dr. Drew, again, I just want to say that you're so easy to listen to and easy to understand. And I appreciate that. And I'm sure everyone else does tonight as well. Linda and Alicia, I want to thank you both too for sharing your obvious passion and your expertise and your experiences with us. So we truly appreciate that. And that brings us to the close of Ovarian Cancer Canada's Research Forum. We hope that you found the sessions helpful and informative, and that, like me, you can feel the passion and the dedication from these experts who are all working towards our shared vision to help people with and at risk of ovarian cancer live fuller, better, and longer lives. So the path ahead calls for added bench strength, and we're confident that our community of researchers, supporters, families, and friends will rise to the challenge. We've honestly come too far to stop right now or even slow down, as you've heard tonight. Thank you for your support and for further progress against this disease. Thank you for your support and confidence in this team. Without you, events like this and progress would not be possible. And with that said, we want to know what you thought about tonight's event. So we would love it if you could take a few minutes of your time to fill in the evaluation form. We are going to link that in the chat right now. And you also will receive a link via email. Your responses really do help us shape future sessions and ensure that we're giving you the programming that's relevant to your needs. Also, I just wanted to remind everyone that we have recorded all of the sessions tonight, and our goal is to have those up on our website by the end of next week. If you have any questions about any of the sessions tonight, please also feel free to reach out to us and we can have our information put in the chat as well. And if you want to learn more about Ovarian Cancer Canada's research initiatives, you can find that on our website at ovariancanada.org/research. And if what you heard today inspires you to further progress, please consider making a donation. Progress is only possible with your support. So again, I want to thank all of the fabulous speakers we had tonight. A huge big thank you to the patient partners in research and the Teal sisters. What a powerful way to get information across and ask questions of our expert, for sharing your wisdom and your lived experiences with us. And thank you also to everyone who attended tonight, our community, for joining us. I want to wish you all the best. Take care and good night from all of us here at Ovarian Cancer Canada.
Yeah. Good night. Whoa.